All right, so today you're going to look at mollusks, and this is actually a really fun lab because everything are basically represented by specimens or doing dissections. So you don't have any slides to look at this time. But one thing, so this phyla is really big, it's really diverse. It has a huge diversity in terms of body size, but also complexity. And from the specimens that we have in lab or have pictures of, you're actually supposed to be able to put them into what are three of the biggest groups within the phylum Mollusca. So this is one of the few phyla where you have to know what are either called subphyla or classes. And so there actually are more than just three within the Mollusca. These are just three of the ones that have most of the extant species within them, and they're also all really derived. So one isn't really more ancient than the other one. And so these include the gastropoda. So those are what we colloquially call snails. And so these have shells, but they just have one shell. And usually they're in a spiral. One of the exceptions that we have in lab are the abalone. They actually don't have spirals in their shell, but they're also within the gastropods. And then the next one is bivalvia, and we have a lot of examples of bivalve shells, and then you're actually going to do a clam dissection. And so, yep, I'll come back to that. And then the next group is the cephalopoda, and those are the squid and octopuses and nautiluses, and you're actually going to dissect a squid. And then the really cool thing, I always make a big deal about how there's a bunch of different ways to be an animal. So you can really compare the inside of the squid and the inside of the clam. And squid are really active predators. They actually have closed circulatory systems. Cephalopods are some of the smartest animals in the world, just by people have actually taught them how to open jars, and they can watch a person open a jar. You can put an octopus inside a jar, and it has figured out how to unscrew it itself by like a completely different mechanism. So these guys are completely amazing, and they kind of blow vertebrates out of the water just by what they're able to learn and do. But both of these groups are sort of equally derived. And so bivalves, first of all, they have a tiny little heart, but it's open at the ends. So they do not have a closed circulatory system, and they're not, we kind of know they're not very fast, right? And so you'll see that a lot of the things you can really easily see in the squid is going to be kind of harder to see in the clam. And a lot of that is the clams have a really reduced coelom. So you'll see a really big coelom in the squid, and the organs are very clear within sort of this hollow body and suspended by mesenteries. And the clam is really using its shells as its skeletal structure. And so it has such a reduced coelom that you can't really see it. It's not easily visualized, but what you will see are that there's gills hanging down the sides, and you're going to have to peel those open and cut the body open to find some of the digestive systems, etc. Also, try to open the clam. What you'll do is actually, I'll draw it up here. You're kind of going to look for this little thing called the umbo. And so try to open it up with the umbo pointing the same way as in your lab manual, because there actually is a front and a rear end, an anterior and a posterior to the clam. It's still bilaterally symmetrical, and it'll look the same as the lab manual if you open it in exactly the same way. Also, for the most part, the clams that we have in lab, they sort of live in the mud, and so you'll find a little tiny foot. And so what they do is they wiggle themselves around in the mud. And that's why water flows into the clam from the top and out through the bottom, and you're asked to find an incurrent and excurrent siphon. In clams that dig themselves really deep into the ground, it can actually be a big extension. And you have, I think there's one diagram that shows you that. But in the ones that are sort of in the in shallow mud, they're a little harder to find. And I think that's it.